mathematical group, and he is also a collaborator meeting Canadian mathematical biology to spend a year as a postdoc in McGill with Michael Mikey's group, and he's also a member of a Canada France collaboration on blood diseases, and he's here to be part of that collaborative effort. So I'm very pleased to have you.
And thank you very much, Emily. That's a great pleasure. The last time I was here was in '99, so it's a big change. I didn't recognize the campus. I don't think you do, but it's very cool. Yeah. Okay. So I'm impressed because it's like the end of the holidays, like only beginning of the year for New Year, academic year, and I'm impressed to see so many people here. So thank you for spending some of your time attending this talk. When I made it, I tried to make sure that it's going to be as entertaining as possible, and so I included some short movies, and I hope you will enjoy this talk. So this talk is a story of what I did in the past almost 10 years now. And it all started at Vanderbilt University in Nashville with Glenn Webb. And it's about the mad cow disease. And what I'm going to say here during an hour is just to tell you the story of what I did on that topic, starting with Glenn Webb. I don't know if it's like the light is too much. Can you read, like, do you read it properly or not? Like, push me back. I don't know. I was not sure. Is it okay?
That's even better.
Thank you. Okay. So the first question I always ask myself when I start to give a talk is like, because you all are mathematicians, is like, does everybody know what is prion? So that's one of the first questions. And so I wanted to start briefly, and I have an hour, so it should be way enough for me, about this old story. Started in France, but when I said started and old, it's 1732, where the first described case of Scrappy was appeared there. And the Scrappy, because we all, most of us here, we study mathematics applied to biology most of the time. We study equations, and we just handle equations and theorems without knowing exactly what is behind these equations. So I wanted to show you, and I will show you some movies or some pictures of the reality behind the mathematics. So this is what happens when a sheep has Scrappy, and it's kind of a nervous disease. And the next short movie will show you, and this is subtitled in French, I'm sorry, but this is how behaves a sheep having Scrappy. Like these have uncontrolled, doesn't have control of the body, and that sheep is going to die within like months. It's very quick. So this is what happened, and that has been described in 1732. Then, in 1936, we see almost 200 years after, two French found the cause. It was a non-conventional agent. We didn't know what it was. Was it a bacteria? Was it a virus? Nobody knew. Until 14 years later, where there was this case called the Crude case. Who in the audience has heard of the Crude case? See, that's why it's nice to remember only one person. So that's always nice to recall this case. So it was in Papua New Guinea. It's close to Australia. I don't have a pointer, but on this is Australia. You go straight up, and there's Papua New Guinea. And oh, there's a pointer. Great. So in 1950, there was a group of scientists going to, at that time, Papua New Guinea was not really explored. And there were a lot of tribes. And one was called the Fore, F-O-R-E, a tribe. And they had some kind of disease, very strange. And this disease was named by the local people, from the Fore people, that was named Kuru, that means shaking of fear. And they thought that was kind of sorcery, and it was not natural, it was so natural. And actually, the women and the kids were touched by this disease, not the men. And they didn't know why, actually. So there were some doctors who eventually got the Nobel Prize, as you said. They started to study this case, which was totally unknown. They never saw something like that before. And they started to, it was 95, to make a decision. I will explain what decision is going to be taken in 1955. And then the number of cases of Kuru decreased until the early 80s. That means that they stopped some kind of process here so that this disease was not spread. And you see the incubation time is very long. It's sometimes almost to 30 years. So that was a disease where incubation time was really long. And you can see some of effects of the disease at that time. That was a release from the 50s. So, as it is written, it was touching mainly the kids and the women, and I didn't know why. And you can see how they were shaking. And actually, the doctors starting to realize that it looked like the case of the sheep of Scrappy. And they started to make the link between these two diseases. I don't know if you understand French. Usually there's a sound in English, but I didn't want to touch anything, so I'm doing French. Yeah, you can read French. And they say that once you got the disease, within a year, you would die automatically. There was no issue. It was not possible to survive cruel. So this is this is the region here. And this is very localized. And they didn't know why. Because it was not, it couldn't be a virus because all the other tribes could be hit. It was not bacteria either, and they didn't know what it was. So they started to study that. And they didn't know whether it was contagious. And in 1960, they found that it was contagious. They tried every animals, like from the snakes to the birds, anything. But what they did, like the experiments all the time would last 3, 4, 6 months. And they realized that was not enough longer. And they need to take more time because incubation time would be longer. So what they did, they waited for a girl. There was a girl who was touched by the disease. They waited her to die. Once she died, they just took a sample of her brain and they flew to Washington, D.C., where they put the sample of the brain to the brain of 2 chimpanzees. And the name of the chimpanzees was famous. It was Daisy and Georgette, and they were just on the paper, the research paper, they put the name of the monkeys. And they waited only 2 years. And after 2 years, I will show you the short movie, they showed that the monkeys would get exactly the same symptoms as the kids. So they say they proved that it was contagious, but they didn't know how. It was not a bacteria, it was not a virus, and it was possible to transmit it to other species. But the incubation time was very long. So when I will talk about this disease, the prion disease, this is one of the effects we are going to say. So when I show you the equations, I wanted to show you what is behind that and the research behind. In the 1980s, there was a medical disaster, growth hormones. Why was it a disaster? Because kids who don't have enough hormones to grow had some injections of growth hormones, but the hormones would be taken from some animals, mostly cows, that was taken from cows and put into the body of the kids, but this way they transmitted the prion and the mad cow disease to the kids, but they didn't know at that time they were doing that. So there was a huge medical disaster in the 90s, but still they didn't know what it was. And finally, in 1982, Stanley Prusiner, who got the Nobel Prize for that, found what it was. I still don't say what it is. This is what it is. It's in our genes. And if you did some biology, you will see. I will explain later what it is. 1985 to 2000, in the 90s, there was the mad cow crisis. In Canada, you were hit a little bit, but it was mainly in Great Britain and France, where you can see in the United Kingdom here, we had a peak in the mid of the 90s, and the mad cow disease started in late 80s, and then in every cattle, when you had a disease in one group of cows, they would just kill all the cows and burn them. So it was a disaster for the farmers. They lost a lot of money. And the reason why there was a peak, and it started in late 80s, it was because they used to feed the cows with animal flour. That means they took the meat and all the bones of dead animals, they just mix it into the powder to get the proteins and feed the cow with the cows themselves. So they were cannibals. And this is exactly what happened for the crew. The crew, they were cannibals actually. And what happens is like the crew, this tribe, every time there was a dead body in the tribe, they would just burn and prepare the body, and the body was eaten by the women and the kids. And the women and kids would eat everything, including the brain and the spinal cord. That means they would just, with only one patient having the mad cow disease, like the prion, they would transmit from feeding by generation to generations, and being cannibal would just be spreading the disease to each other. And it was not a virus, because it was just by eating the brain of the other people dying. And the men would not eat the dead body. It was only women and kids. That's why it would spread.
I understood they ate the meat but not the brain. They ate the muscle but not the brain.
Yeah, they would eat also the brain. Actually,
No, but the men would eat the muscle, the women would eat the brain.
that's what I read. But actually, this documentary, I will put the reference online if you want to. It's a 52 minutes documentary about the crew. And actually, they said they would like mainly eat. It was only women and kids. That's what I read too. But in the documentary, that's what they said. So I don't know. But actually, this documentary, like from the movies, like the short movies that I showed you, is coming from, is very documented. And actually, there are like some witnesses of that time, and they said like there are like people from 70 years old, and they said like back in that time there were kids, and they would say they would love human meat. It was like tasting like chicken or something. So you can see in the documentary they say what human meat tastes like. So, but this is something that they realized. And once they realized that because it was cannibalism, in '55, the medical doctors there, they decided to stop this cannibalism, and it was forbidden to eat each other. And so it was, they stopped in '55, the cannibalism, and this is how you can see this decrease. And this is what happens exactly for the mad cow disease. They used to feed with these proteins coming from cows, before, I studied it back in the 70s, but before they would just kind of give a treatment to this flower or these proteins to eradicate these agents. But they didn't know they would eradicate that. And then it was going through some processes, and to make more money, they decided to just don't do a process that was for them not kind of cleaning process that would that would save money for them. And just by removing this process, they added the prion to the mad cow. And then in the late 80s, they started to give the prion without knowing it to the cows. And in the mid 90s, they decided to stop to feed cows with these proteins, with the cows themselves, the cannibalism. So that's why it's all started to decrease, and right now it all stopped. The protein, we call this animal protein, has been stopped. And you can see some of the effects of the mad cow in 95. And what they do, the same way it was doing some effects to the crew, it was exactly the same as Crepy and mad cow. So what they did is just like they did to burn. Every time there would be a cow dying of this disease, all the other cows would be killed and burned. So what happens, like they fed the animals with cows, dead cows bodies, and that would be transmitted to the veal and the cows, and we would just do that again. The question was, is it transmittable to human, like it was contagious to chimpanzees? The response was yes. And it started at that time to have some cases of what is called Creutzfeldt-Jakob disease. And at that time it was a major crisis because some British people started to be sick of that. So when I'm talking about this disease, this is the effects of the disease I'm talking about. That means once you get the symptom, it lasts only 1 year before you die. And you have no other issue. It's like for human, for sheep, for cow, for everyone. And you can see a normal brain here, and you can see a brain having this disease, the Creutzfeldt-Jakob disease. So it's called the bovine spongiform encephalopathy, and it's BSE. Why is it called spongiform? Because the brain looks like a sponge. In humans, it's called the Creutzfeldt-Jakob disease. You have it comes from Creutzfeldt and Jakob, and depending on the disease, it touches several parts of the brain. You know, crew, for instance, is this part of the brain. Creutzfeldt-Jakob is this and this, also here and here. This is a variant. This is another kind of same disease, and this is fatal insomnia disease. That means that it's the same process. People having this disease will die because they are not going to sleep anymore. And it lasts only 1 week. After 1 week, if you don't sleep at all, you just die. So, going back to the question, what is prion? It's a protein. It's not a virus, it's not bacteria, it's just a protein. And in the normal form, on the left, here, it's called prion protein common or similar. It's called PRPC. So every time I will talk about PRPC, it's the normal form that everyone here produces in the body. And you see there are like some red helix here, and blue, what is called beta sheet. And in the pathological form, you have prion proteins, crappy, and it's called PRPSC. And what happens? What is the difference between the left and the right? This is the same protein, but the helices started to form some beta sheets, and these beta sheets are going to be the link between each other, and they are going to get some polymer. So when you don't have, when it's a blue form like that, they cannot attach to each other, which is called a monomer. When it's getting bad, like this red form, then it's going to attach to each other, it's going to be a polymer. And this is the key of our model. And then once you know that, just to understand the model is going to be easy, the mathematical model. Just to show you the size comparison of a cell, here you've got the nucleus, you've got the bacteria, you've got the virus, and this would be a protein. Just to study that from a biological point of view, it's not almost impossible. It's very, very hard to study that. It's different to study a protein than studying a virus or a bacteria. And in the brain, you can see when you emphasize this in the microscope, they make some hole in the brain, this sponge form of the brain, and close to these holes, they just make some aggregates, and these aggregates are called protein-prion plates. Okay, so these are the long chain of polymers of prion, being some kind of fibrils, and these fibrils make some plate like that. So now I can, like, the biological feature has been set up. I can start with the model and the mathematics. And it all started in the early 90s, where Peter Lansbury made some hypothesis of the formation of this prion. And you can see the good one, the blue one, the PRPC, is going to unfold to get this kind of beta sheets. And once it's in, and this can go back, so it's reversible. But after a certain point, it becomes more and more able to attach to other protein, and then it starts to make a chain. And these chains once make kind of a loop, and it's going to be stable. And once it's stable, you have only one way. It's not reversible anymore. And then the disease starts. Before, it's still possible to go back. And I'm sure that in our brains, in our life, we can go up to this step. What is dangerous if once we go there? Because then you have an incubation time, and then we get the disease, and then we die. Which happens sometimes. It can be sporadic, it can be like spontaneous at around 70 years old. So what happens, like when it's a nucleus, that's the nucleation form. This is the nucleus. It becomes like a loop, which is stable. Then it starts to polymerize, to grow, and then it can break sometimes. And when it breaks, it goes back to some reversible process. But I will tell you more in the next slide. The next slide is the first mathematical model we studied back in 2003 with Glenn Webb and Meredith Urrea. And that was our model. I will show you the model and then the equations after. So we started with the good one. The monomers here, they can be degraded at some rate. They can be produced by our cells because it's a protein, so it's produced by our genes. And then what happens if we have a prion injected in our body or already existing in our body? Then this long chain of polymer can split into two ways, either two big branches or one big and one small. And if it's smaller than the nucleus, it comes back to the good one. See, that's the process of the splitting. Or the other fate, it can lengthen by adding a good one. So a polymer can split or lengthen. And when it splits, it can split into two big chains, or one big chain, and or another part going to the monomers. So we decided to put these dynamics into equations. And these are the first equations we set up in almost 10 years ago. So this is for the monomers. The monomers have no size, so it's only differential equations. So it's only depending on time. So you have the source, which is the production here. You have the degradation, which is here. And you have here the loss of monomers that are going to be attached to the lengthening here. And the big part here is the fact that it's the monomers coming from the splitting here. So you have U is the number of polymer, X0 is the size of the nucleus, and under this size it's going back to it. If you want some more details, I can give you the papers if you want more time to read the equations. And for the polymers now, they are structured in size because the polymer has a size, and the size is X here. So you have hyperbolic equations. So it starts with the transport here, depending on the number of polymers, and you have the degradation rate, which is here, and these are the one coming from the splitting of two bigger sites. Okay, and so here is the splitting rate of here. Oh no, sorry. The splitting rate is this one splitting, so there's the loss, and the gain is a bigger size splitting with the size of X. Is it clear? I hope so. So we decided to make some simulations and see what was going on. And I will tell you about the mathematical results after. This is some of the simulations we did. So we put, this is the time here that goes from 0 to 20 days. This is the polymer length that goes to 500 monomers inside, the size of the polymer. And we started with the initial condition with the Gaussian curve here. And what we see is it all started by lengthening a lot. If you just see the mean length, it's increasing here. And then at a certain point, it goes back here. And the big length becomes shorter here. And it starts to stabilize here. So we decided to see this in a longer time scale. So instead of 20, it was 200 days. And what is the point of the arrow here is this square. This cube here is supposed to be here, 20 days. So you see it's growing very high, and it starts to stabilize. The question we had was, why is it lengthening with such a great length at the beginning, and then goes back here and stabilizes? What is this process? So that's what we were wondering. And the answer came very naturally. Let's say that we are in the cell, or the body, let's say, and each of us are the initial quantity of good prion. So we are all monomers. And we introduce a polymer inside the room. So what is going to happen? We are going to attach to this polymer. So each of us are going to attach. They are going to split, to attach, etc. And because we are numerous here, enough, we are going to build long chains. But after a certain while, the number of monomers of us single are not going to exist anymore because we are just attached to everything. So we are not going to lengthen anymore. What we are going to do is split. So the mean length is going to decrease because of the splitting. And this is why we start to lengthen. And because of the lack of monomers, we are going just to split and get an equilibrium of the mean length. So that was an explanation of this process. So we decided to compute it, the mean length, and you can see that it increases the mean length of the polymers up to a certain threshold, and then decreases because of the lack of monomers. And then it doesn't go to zero. We just proved there was a positive equilibrium. So it goes to a certain number. This is due to the fact that there was a production of monomers. The lambda was a production of monomers. So these are the results, mathematical results we went through. We made some assumptions so that analytically it was possible to get some results. So the beta x, which was the splitting rate, was linear, beta of x. Mu of x, the equation was constant, and the kernel of the splitting was the probability to split was uniform. You can split anywhere on the chain. Like practically, it would be more in the center of the chain, but we took it as uniform, like anywhere. And we decided to use the integral of the total population of the polymers. D of t is the monomers, and P of t is the total mass of the polymers. That is the number of monomers in polymers. So it's the integral of x times Q of x. With that, we integrated with respect to x, and we got a system of 3 equations. And for those who study epidemiology, this looks like a SCMAI model. We were able to find out some existence unit test positivity, which was not very difficult. This is very classical. So it was there was a unique solution for the initial problem with a positive form. So that was no problem. And we showed there were 2 steady states. Do you have a question? Oh yeah. And one was the disease-free steady state. That means that there is U and P are related to the polymers, the bad prion, and V, which is in the middle, is related to the good one. That is, the equilibrium here is where we eradicate the disease, and the second one is where the disease is not eradicated. So for the first one, it was possible to show that under this condition here, kind of a R naught for the epidemiology, it was possible to get this stable, totally stable. And for exactly the other one condition, it was possible to show that this one was locally stable. So there was kind of transcritical bifurcation. That was not difficult there with classic results. And what we were able to prove that was not shown before was the global stability using Lyapunov functionals. It was not easy to find, but we found one. And actually we could apply that to this epidemiological model too. So in the same paper, we wrote the solution for this disease, but also to some epidemiology paper. So it was 2 things into one paper, and with some simulations to show that it was globally stable under this condition. After that, many other papers came out because some were very mathematics papers with studying only the PD equations and not the 3 differential equations. I'm not talking about that anymore because when I came back to France to get this position in Lyon, I was contacted by a biologist in Montpellier. Montpellier is southern France. And this biologist came to me and said, I read your paper, which is good for a biologist because it was mathematics. First, it was good point for me. And the second one is like, when I do my experiments, I found almost exactly the same curves as you. First, I was happy, but for him, it was almost, which was not good. And I will show you why. The first things he told me about this our model is that there were two main issues. The first one, we already, in our model, already set up this polymer inside the cell. That means the nucleation process was not modeled in our, that means the spontaneous case will not appear. We studied that with one of my PhD students, and it's more stochastic. This nucleation process is very stochastic because every process is reversible, so the probability to get the first case is very stochastic. And then once the first case of nucleus appears, then it's deterministic, and we just studied the deterministic part. I will not show you the this nucleation process. This is not part of my talk, but if you're interested in, I can show you after. I'll just put some references on that later on. One of the most important problem we got with the biologists was what we call the time lag. The lag time, sorry. The lag time, these are our simulations. When we inoculate virtually into the brain of the mouse concentration of prion to the brain, the more you inject a big concentration of prion, the more the disease is going to appear quickly. Like, that means that the onset of symptoms is going to be, let's say, you inject inoculum, the concentration is here, and the days of onset of the disease is here. So if you inject a small quantity of prion to the brain of the mouse, you would have like 200 days before the onset of the symptoms, and then the mouse is going to die. Then the higher you inject the concentration of prion in the brain, the less days are going to be necessary to get the onset of the symptoms. And in our curve, what we did is just it was going down to 0. So if you inject a high concentration of prion to the brain of a mouse, almost immediately the mouse is going to get the disease and the onset of the symptoms. So this is
so does this mean that you inject the prion the same time, or
a high dose of prion? Yeah. Actually, that was our simulations, and this was what the biologists did after our model. And so you see, this is the variance is pretty high because this is the nucleation process at the beginning. So he injected high concentration of prion into the brain of mouse, and he got something, but after some time it didn't go like if you make. I will show you. Maybe I have a clearer. Instead of having something going to 0 straight like that, he got something with a threshold. And what we did with our simulations is like the higher we go to the concentration, the more we go to 0 here. So instead of having a curve like that on the onset of prion, it would just start immediately. And so what he got here, biologically speaking, is like after the onset, after the injection, he would just have some times before getting a highest level of prion, and the more you add the concentration of prion, instead of starting like directly, he would just stop here like a wall. Like that means that even if you inject a high quantity of prion, you have to wait a certain time that you cannot decrease. Like there's a certain lag time you have to wait. And this in our model we didn't have that. As I showed you before, it was starting immediately. So there was some discrepancy